This is Big Idea 4. It reads biological systems interact, and these systems and their interactions possess complex properties. Now this is 2020, and we are not covering, on the AP Bio exam, we are not covering certain systems, and they removed the entire ecology and evolution units. So this video is cut short, where I'm only presenting you the molecular and the cellular interactions that we find in biological systems. So here we go, molecular. Okay, so this is a review of some of the biochemistry. Remember that you have monomers that are joined to form polymers, which make up your organic compounds. Remember, you have dehydration synthesis, which is a process that removes water, so an OH on one side and a hydrogen on the other, removes that and forms a covalent bond to join monomers. So you have a string of nucleotides that will form your nucleic acids such as DNA and RNA. The monomer or nucleotide, the nucleotide monomer consists of a sugar phosphate backbone. It alternates, and then it has a nitrogenous base in the middle. Keep in mind, if we're talking about DNA, it's double-stranded, and the two strands go anti-parallel. Carbon, hydrogen, oxygen, and nitrogen. Remember, nitrogen is part of the nitrogenous base. Phosphorus is part of the phosphate group. These are the elements present in nucleotides. Now, if I wanted to break down nucleic acids into nucleotides, we go through the reverse process called hydrolysis. So that's the addition of water. You add an OH here and an H here, and then you're breaking the covalent bond. Okay, so dehydration synthesis is joining monomers. Hydrolysis is to cut, is to break monomers. The next group of organic compounds are your proteins. Monomers are called amino acids. So let's quickly review composition of amino acids. You have a carbon, you have a carboxyl group, and an amino group. And then you have a middle R group, and there are 20 different R groups. So there's 20 different amino acids, and they can be categorized based on their properties. So you have hydrophilic or polar R groups. You have hydrophobic or nonpolar R groups, and they tend to, all those R groups that are nonpolar tend to be near the core, away from water. And then you've got your ionic R groups, which can be acidic or basic. Okay, so you have these different R group interactions. The elements present in amino acids are carbon, hydrogen, oxygen, nitrogen, which is part of the amino group, and certain R groups like cysteine have a sulfur in their R group, and the sulfur of another amino acid actually form a disulfide bridge, which is a really tight linkage. It helps stabilize the structure, the tertiary structure of proteins. Okay, so let's talk about protein structure. The primary structure of proteins is determined by the amino acid sequence. And remember, amino acid sequence is determined by your nucleotide sequence. So if there's a change in the nucleotide sequence, there may be a change in the amino acid sequence, depending on where it happens. The secondary structure of protein is determined by hydrogen bonds. The hydrogen bonds form between the amino group, that's slightly negative, and the carboxyl group, the amino group is slightly positive, and the carboxyl group is slightly negative. There's a slight attraction between those two polar opposites, forming weak hydrogen bonds. This leads to formation of alpha helices or beta pleated sheets within the secondary structure of proteins. Tertiary structure is determined by R group interactions. So like I said, nonpolar and nonpolar are going to be near the core. Hydrophilic are going to probably be more outside, near water. And then you have your acidic and your basic, your positive and negatives interacting as well. And some proteins that are made up of several polypeptides will have a quaternary structure where the polypeptides each kind of come together based on the interactions on the side chains on their surfaces. Okay, like hemoglobin has 1, 2, 3, 4 polypeptides, and they all come together to form the overall hemoglobin protein based on the quaternary interactions. All right, fatty acids. Fatty acids form your lipids. Fatty acids are pretty much nonpolar. They have a lot of carbon and hydrogen bonds. And if you only have single bonds within your fatty acid, you're considered saturated. And these make up your solid lipids because of the tight associations between the carbons and the hydrogens, the tails. The nonpolar tails tend to stick together more closely, as opposed to unsaturated lipids that have double bonds, and the double bonds form what we call kinks. So there's like a slight bend in the tail of the fatty acid, which allows for greater movement between the tails. So these are typically your liquids at room temperature and increases the fluidity of your membrane. Okay. Monosaccharides are your sugars. So they join to form carbohydrates, and typically you have them in this ratio, carbon, hydrogen, oxygen. Glucose, for example, is a monosaccharide. And you can string glucose together, and based on the way that you string them together, you can have your starches, or you can have your glycogen, or your cellulose. Okay, so let's talk about another interaction that's really important in biological systems, and that's the interaction of enzymes and their substrates. Okay, interactions between molecules affect structure and function. Okay, so let's take a look at some of the factors that affect enzyme activity. Interaction affected by substrate concentration. It makes sense, right? If you have more substrates, the enzyme is more likely to find the substrate and bind it to the active site and be able to convert it. The environment. 3 different conditions in the environment can either increase or decrease the rate of reaction: pH, temperature, and ionic conditions. So every enzyme has an optimal environmental condition. If you change that condition, it's most likely going to interfere within the protein structure and cause certain bonds to disrupt. And if that happens, the active, especially in the active site, if that interaction shifts or changes, the substrate is going to have a harder time binding. Competitors. The addition of competitors can decrease enzyme rate. For example, you've got competitive inhibitors that actually mimic the substrate, so that instead of they're competing for the active site, so here's a substrate, here's a competitor. If you have more of the competitors, they're more likely to bind to the enzyme. Non-competitive inhibitors are inhibitors that find the allosteric site of the enzyme, and by binding to the allosteric site, which is a different site from the active site, it actually affects the active site. So it changes the shape, and by changing the shape, the substrate can't bind. We also talked about cofactors and coenzymes. These are factors that actually increase the activity of the enzyme, usually because they make the active site a better fit for the substrate. All right, moving on to cellular interactions. Know your cell parts. Also know the difference between prokaryotes versus eukaryotes. Remember the evolution of eukaryotes from prokaryotes. There are 2 major hypotheses. The autogenous hypothesis states that the membrane of a prokaryote infolded or folded in on itself, creating some membrane-bound organelles. And then we have the endosymbiotic hypothesis that tries to explain the origins of mitochondria and chloroplasts. So you've got a small prokaryote being engulfed by a large prokaryote. The small prokaryote had the ability to convert food into energy, or ATP, for the larger prokaryote. They both kind of benefited each other and existed in symbiosis. Okay, that must have happened first because remember, plant-like organisms have both mitochondria and chloroplasts. So a few of that group that evolved with the mitochondria then went through a second endosymbiotic event where a prokaryote that's photosynthetic was engulfed. Okay, structure relates to function. This is a very important theme all throughout biology, so let's tie it into cellular biology. So anytime you're looking specifically at a type of cell that performs a specific function, you're going to find structures that help it in that function. So white blood cells and endocrine cells, for example, you would expect lots and lots of endomembrane system organelles because they're producing antibodies, which are proteins, or hormones for endocrine cells. So in order to produce lots and lots of proteins and secrete it outside the cell, you're going to need the rough ER, you're going to need the Golgi, and ribosomes, lots of ribosomes, right? White blood cells also probably will contain lots of lysosomes because some white blood cells engulf foreign invaders and the lysosomes chop up through hydrolysis, chop up those invaders. Okay, muscle and sperm cells, they need lots of energy. So it makes sense that if you look inside a muscle or sperm cells, you're going to have a high concentration of mitochondria. Interestingly, red blood cells, their main function is to carry oxygen and carry lots of it. So in order to maximize the volume and the space for oxygen, the red blood cells, as they mature, they actually spit out their organelles. They spit out the nucleus. That's why they only survive for about 3 months. And they make room for hemoglobin. So red blood cells are unique because they don't really contain much of all the other organelles that other cells have. And that's to make space for hemoglobin so that hemoglobin can supply oxygen to all the cells in the body. Okay, that's it for today.